All right, we're still chapter 43. We're looking at adaptive immunity and the receptors that provide pathogen-specific recognition. There's a couple of terms we need to know here. Lymphocytes. These are going to be white blood cells. If they mature in the thymus, then it's called a T cell. If it matures in the bone marrow, then it's a B cell. All of these originate from stem cells in the bone marrow. There's actually one more kind called a natural killer cell. We're not going to talk much about it today. The other things we need to know, I think we've already done antigen, anything that gets a response from your immune system, and exposure to a pathogen activates B and T cells with antigen receptors. And these are specific for parts of that pathogen. The small accessible part of an antigen that binds to the antigen receptor is called an epitope. So that's the accessible part of an antigen that binds to an antigen receptor. B and T cells have receptor proteins that combine to form molecules, and each individual lymphocyte is specialized to recognize a specific type of molecule. All right. Each B cell antigen receptor is this Y-shaped molecule. It's made up of two identical heavy chains, what I just outlined, and then two identical light chains. And then you have these C regions are constant regions and B are variable. The variable regions are what provide antigen specificity. The binding of a B cell antigen receptor to an antigen is an early step in B cell activation. This gives rise to a protein. This gives rise to cells that secrete a soluble form of protein called an antibody or immunoglobulin, abbreviated IG. Secreted antibodies are similar to B cell receptors, but they lack the transmembrane region here. They're just not attached. Antigen recognition by T cells. We have each T cell receptor consists of two different polypeptide chains called alpha and beta. Again, you have your constant regions and variable regions. The tip is the variable region. The T and B cell antigen receptors are functionally different. I assigned some videos. Well, I sent you a link to some videos on the McGraw-Hill website, and then we're going to watch a few animations tomorrow in class as well. And some of this should make more sense when we see it in action. But the T cells bind to antigen fragments that are displayed or presented on a host cell. These antigen fragments are bound to cell surface proteins called, just kidding, where'd I go? Hang on. Okay, I got lost, but I'm back. These antigen fragments are bound to cell surface protein called MHC, which is multiple. No, I'm sorry. Undo. Major histocompatibility complex. The MHC molecules are host proteins that display the antigen fragments on the cell surface. So you can see in infected cells, if you look here, you have the MHC molecule here binds with antigen fragments and transports them up to the cell surface in a process called antigen presentation, which is presentation. You know how I abbreviate. This is necessary for the T cell to be able to participate in the adaptive immune response.
Do I have one?
B cell and T cell development. Geez. The adaptive immune system has 4 major characteristics. Diversity of lymphocytes and receptors is number 1. Number 2 is self-tolerance, and that's the ability not to attack yourself. Number 3 is B and T cells proliferate after activation, and 4 is immunological memory. So, these are our 4 major characteristics. When we look at diversity here, by combining variable elements, the immune system assembles a diverse variety of antigen receptors. The immunoglobulin gene encodes one chain of the B cell receptors. Many different chains can be produced from the same gene by rearranging the DNA. Rearranged DNA is transcribed and translated, and the antigen receptor is formed. Let's look at a picture real quick. So this is showing how, wow, there's a lovely line. Hang on, that's from last recording. How you have DNA, how you get from your DNA. Oh, I don't have a marker. Sorry. Down to your actual variable and constant regions. Self-tolerance. Antigen receptors are generated by random rearrangement of DNA. So as lymphocytes mature in bone marrow or the thymus, they're tested for self-reactivity. Is this going to attack yourself or not? Some B and T cells with receptors specific for the body's own molecules are destroyed. That process that it's destroyed by, let me see if I can get a pen, is, jeez Louise, apoptosis. just programmed cell death. And then the rest are rendered non-functional. Proliferation of B and T cells. You have a few lymphocytes with antigen receptors for any particular epitopes. In the lymph nodes, the antigen is exposed to a steady stream of lymphocytes until a match is made. And this binding of a mature lymphocyte to an antigen initiates events that activate the lymphocyte. Once activated, your B or T cell undergoes multiple divisions. The proliferation of lymphocytes is called clonal selection. Multiple division of lymphocytes. Two types of clones are produced: short-lived effector cells. They have an immediate response, which is good since they're short-lived, right? Against an antigen. And then your memory cells give rise to effector cells. Give rise to effector cells. And it's if that same antigen is encountered again. And so these are long-lived. That says lived, not long. I just love this so much. All right. And, oh, dang it. Hang on. Immunological memory is responsible for long-term protection against diseases, either due to prior infection or vaccination. The first exposure to a specific antigen presents the primary immune response. During this time, selected B and T cells give rise to effector forms. And then the secondary immune response, you have memory cells that facilitate a faster, more efficient response. So next we just have a couple of pictures. This is showing your B and T cells, how you're going to end up getting your memory cells and plasma cells. And then primary response versus secondary. The end. Good night.